In this lecture, what I like to look at here is how the cell theory was developed based upon a historical overview. And so what we're going to look at are the contributions of some specific scientists and how their evidence was put together finally into something called the cell theory. Our first person that we look at here is a gentleman by the name of Robert Hooke. Hooke in the 1600s, 1665 to be specific, was looking at slices of cork under a very primitive microscope. Now, cork is the outer layer of the bark on a tree called the cork tree. And when he looked at it, he found that the cork, here's an example of what it would look like, was composed of these tiny little units that resembled the honeycombs in a bee hive. And he called them cells. They resembled something that he knew in his experience to be a cell, a unit. And at the time, cells were primarily prison cells. And he referred to these as cells. And again, what he was actually looking at was not a complete cell, but the remains of a cell after it had disappeared. And since cork is found in a plant, trees are plants, what he actually saw here was the outside of a cell, a plant cell, and that would actually be the cell wall, because that's usually what's left. All of the other cellular organelles and components have long since dried up and blown away in this. The next contribution in 1680 was van Leeuwenhoek. Van Leeuwenhoek is Dutch, and he was looking at pond water samples in a very simple microscope. And what he observed here was living cells for the first time. Now, not knowing specifically what he may have been observing, he was observing such as amoeba, or could be in paramecium. In a lab that we will do, hopefully, you will actually get to do what van Leeuwenhoek did and take some pond water samples and see what you can find within them. So van Leeuwenhoek was the first to observe living cells. Then the next contribution was several hundred years later, 1830, because what happened here is we had the advancements of technology. We were able to create a microscope very similar to what you will be using in class, and the person that was able to observe the next contribution was actually to observe a organelle within the cell, and that would have been Robert Brown in 1830. He was looking at living cells, could have been a cheek cell, and he was able to stain it and observe for the first time that within the cell there were structures, and they were referred to as organelles, similar to organs within a larger organism, a human body, for example, or any kind of animal. This is also one of the labs that I hope that we'll be able to do in the hybrid situation, where you will actually be able to stain and observe your own cheek cells. As a matter of fact, you will be creating a video of this for me. The next contribution was by a gentleman called Matthias Schleiden. Schleiden in 1838 was observing plant cells. He was a botanist. And he was able to observe that all plant cells, no matter what, he was observing different tissues from, say, the leaf, the root, the stem, the flower petal, etc., that they were all, by observing them under a microscope, composed of units called cells. And similarly, a zoologist by the name of Theodore Schwann, he was able to observe in animals, observing different tissues of animals under the microscope, that animals were all composed of cells. So he observed muscle tissue, neural tissue, connective tissue, etc. And under the scope, he was able to see that they were all composed of these units called cells. Again, in this 1800 time frame, because technology had advanced to the point where we could do this. final contribution was by a gentleman called Rudolph Virchow. Virchow was able to observe the first time a cell actually reproduced. He was observing some living cells, probably pond water, and he was able to observe that a cell divided in two. Here's an example here of a cell that is undergoing a stage called mitosis. If you remember from your sophomore biology, that would be the stage called anaphase. But he was able to see that, not knowing, I don't know specifically what cell he actually observed dividing, but he was able to see and observe reproduction for the very first time. And again, this was during the 1850s. So that 1850s, 30, 40, 50 time was a very prominent scientific discovery. And it was due to the fact that we had technology that allowed us to do this. The microscope that was developed at that time was very similar to what you guys will use in class. So from the contribution of all of these gentlemen, a theory was developed referred to the cell theory, and it is composed of three parts. Okay? First, all living things are made up of cells. They're either composed of multicells or a single cell themselves. And that cells are the basic unit of structure and function in living things. So the cellular level is the basic unit of where life would begin. And finally, that only cells can produce other cells. That living cells can only come from other cells, the process of reproduction. So this would be our cell theory. And it is based upon the contribution of these different gentlemen, from Robert Hooke naming a cell all the way down to Rudolph Virchow, who actually observed that cells reproduce. They just don't spontaneously pop up out of the ground. So based upon this, you will have an assignment in your OneNote to complete, and you will see that on the page underneath the lecture, for you to summarize and outline the contributions of these gentlemen. You will need to know who they are, what they do, and what contribution they had to the cell theory, as well as what are the three parts of the cell theory. So after finishing this, you can move on to that assignment, submit the checkpoint that you'll find, and submit for it.